Hello everyone. For this Module 1 discussion, I'm going to briefly go over a study which analyzed two separate catalogs of human genome variants. One completed by the International HapMap Consortium cataloged known common variants, while the other initiative, the 1000 Genomes Project, conducted a pilot study which collected genome sequences of 2,500 individuals to identify rare variants, including those with a minor allele frequency of less than 1%. So when the data from these two resources were compared, the results showed that approximately 72% of the HapMap single nucleotide polymorphisms, or SNPs, were also found by the 1000 Genomes Project pilot data. When variants with a mutation allele frequency of less than 5% were excluded from the comparison, however, the overlap in SNPs between the two resources jumped to 99%. So when classifying this study, in my view, this was a meta-analysis under another type of research category. One can certainly argue that this would be a quantitative observational descriptive study, which was my original instinct to categorize as such, but that leads me into the challenges of labeling this type of study. There was feedback from my team that this should be listed as observational descriptive study, which had been my original thought, but after listening to the lectures from Professor Scotch, I felt that it did not quite fit that traditional mold to be clearly listed as such. This meta-analysis between two large biomedical datasets is utilizing existing data and using the method of synthesis to show an overlap or lack thereof of this data. This is a challenge when establishing the study classification, since there is a sort of natural temptation to fit something perfectly into a box, but as Professor Scotch mentioned, sometimes it won't necessarily fit within that descriptive study all the time, and oftentimes there's not a correct answer to where it fits into one of these categories.